Hi, we are going to talk about the spinal cord. The spinal cord is located in the vertebral canal. That is the succession of the vertebral foramina. The spinal nerves are living through the intervertebral foramina, the ones you see here. The spinal cord in a mid-sized dog gets up to the level of L6, L7. The external morphology. On the superior image, you have a craniotomy, a dorsal craniotomy, and a dorsal laminectomy, where you can identify the encephalon inside the cranial cavity and the spinal cord located in the vertebral canal. We also identify the nerves that form the brachial plexus and the lumbosacral plexus. On these images, we identify the central and peripheral nervous system. I say central and peripheral because the central is formed by the encephalon and the spinal cord, and the peripheral nervous system is formed by the roots of the cranial and spinal nerves. You see that the spinal cord is a cylinder-like structure, is a very long one, and has enlargements, two enlargements that correspond to the cervical intumescence and the lumbar intumescence. The spinal cord is enlarged in these places because there are more neurons, there are more motor neurons that are going to form the brachial plexus and the lumbosacral plexus. The spinal cord can be divided in sections. The first one is between C1 and C5 spinal segments. Remember that there are as much spinal segments as vertebrae, with one exception, is for the cervical level, because the cervical spine is made of 8 cervical spinal segments, while there are 7 cervical vertebrae. This means that the spinal nerves are going to live at the cervical level rostral to the vertebra of the same number. Then at the thoracic, lumbar, sacral, and caudal levels, the spinal nerves are going to live caudal to the vertebra of the same number. So the second segment is between C6 and T2 and corresponds to the cervical intumescence. The third segment is a very long one. It runs between T3 and L3 spinal segments. And the last segment is between spinal segments L4 and S3 and corresponds to the lumbar intumescence. The spinal cord is surrounded by membranes. They are called meninges. The term meninge comes from the Greek and it means membranes. And here you see the spinal cord and the meninges. In this case, we have done a dorsal section in the meninges. So we identify the meninges, okay, and it has been removed, the meninges from the spinal cord. So the meninges are 3. It's 1 pachymenenge and 2 leptomeninges. These are the arachnoid and the pia mater. So there is 1 dura mater here. This is the more external one. Underneath we have the arachnoid, and then in close contact with the parenchyma of the spinal cord, we have the pia mater. These are the leptomeninges, and this is the pachymenenge. Pachy meaning thick, and lepto meaning more thin membranes. Okay. These meninges surround the spinal cord and the roots of the spinal nerves. Here in this image, you see the dura mater is the outermost membrane or meninge. Underneath we have the arachnoid, and in close contact with the parenchyma of the spinal cord is the pia mater. In between the arachnoid and the pia mater, there are connections. So this space here, it is called the subarachnoid space. So as you see here, it is important to notice that the dura mater and the arachnoid are covering the spinal cord, and then they also cover the proximal portions of the roots of the spinal nerves. And then, as we progress most distally, so each root, the dorsal root or the ventral root, are covered by a single sleeve of dura and arachnoid, and also the pia mater in close contact with the root of this spinal nerve. Here you have a video where there is the encephalon and the spinal cord covered by the meninges, and the outermost meninge is the dura mater, the more thick one. As you see, the meninges also cover the roots of the spinal nerves. Here is at the level of the cervical intumescence, the brachial plexus, and then we get to the thoracic level. Here, the roots of the spinal nerves are shorter. Now, when we get to the lumbar level, here we have the lumbar intumescence, okay? And then as the spinal cord is shorter than the vertebral canal, the roots have to be longer in order to live through its proper intervertebral foramen or the sacral foramina. What it happens is the dura mater and the arachnoid, they are covering most of these, in a single sleeve, most of these portions of the roots of these sacral and caudal spinal nerves. This is a case of a cat. Here we haven't opened the cranium, but we identify the spinal cord in the vertebral canal. You see it is still covered by the meninges. And as you see here, there is not much space in between the spinal cord and the bony structures that form the vertebral canal. So that means that any process that is occupying this space is going to damage first the softer tissue that is the spinal cord. So now we are at the thoracic level, and then we progress to the lumbar level. We are at the level of the lumbar intumescence, and here you will notice that the spinal cord of a cat is longer than the spinal cord of a dog. It reaches up to the sacrum. So this image is very important to identify what I have said before, that the dura mater and the underlying arachnoid are forming a single sleeve for the spinal cord and for the roots of the spinal nerves. However, when these roots of the spinal nerves are longer, we clearly identify something, that the dorsal root and the ventral root are surrounded by a single layer of the dura and arachnoid. They form a single sleeve, one for the dorsal root and one for the ventral root. This is the dorsal root with the spinal ganglion, and then this is the spinal nerve. This is why when on a transverse sections on MRI, these are T1 enhanced sequences, you see the spinal cord here, and then you see the dorsal root of a spinal nerve and the ventral root here as well. the dorsal root, and the ventral root. Sometimes, if you are lucky, you can even identify the spinal ganglion in the dorsal root. And what do we see here? As it is a T1, there is hyperintensity. The signal is hyperintense here. And that means that there is epidural fat in between the roots. This is the epidural space. Let's go with a video. In this dissection, I have removed the dura mater and the underlying arachnoid membranes. So here you have the dura that has been sectioned. So it was covering in a single sleeve or layer the spinal cord and the roots. These are small roots, rootlets of the a dorsal root of a spinal nerve as well. The same happens in the ventral root. And as you see here, there is the dura mater section and the arachnoid underneath the dura. And here there is a space, because then, as in the distal portions of the roots of the spinal nerves, these membranes, they form a single sleeve for the dorsal root and a single sleeve for the ventral root. In this case, we are at the level of the, brachial plexus. And as you see, I am putting the forceps in between the dorsal and the ventral root. So this is to show you that here there is the single sleeve for the dorsal and a single sleeve for the ventral root. So we may find epidural fat at this level. Now you see the dura here. You see how the dura then is going to form a single sleeve for the dorsal and a single sleeve for the ventral. However, at the proximal portions of the roots of the spinal nerves, they surround the spinal cord in a single layer and a single sleeve. So again, this is again from a dog, is from the caudal cervical level up to the caudal level. A dorsal laminectomy has been done here. We see at the level of the brachial plexus. So here the spinal cord is surrounded by the meninges. We see the epidural fat. Epidural fat here. So we are at the thoracic level and we are progressing to the lumbar level. Here you have the lumbar intumescence, and as you see, this surrounding gets inside the sacrum and progresses up to the caudal level, attaching to the caudal vertebrae, this dura. So what did happen? It happened that, as I said before, the spinal cord is shorter than the vertebral canal. So that means that the roots of the spinal nerves, the last lumbar, the sacral and the caudal spinal nerves, they have to be longer. This is the lumbosacral plexus. And these roots form what is known the cauda equina. You see the roots here? They are quite long. This is L6. However, more dorsally, the roots are shorter because they live through the intervertebral foramen that is closer to the origin of the spinal nerve. Okay, we have talked about the coda equina. Here in this image, you see clearly this is an enlarged area of this rectangular area has been enlarged. And as you see, it is surrounded by the meninge, by the dura mater, and the rest of the meninges. And as you see, these roots are very, very long. So they form the coda equina. In this image, there is a dissection where what I have done is to open the dura and the underlying arachnoid, and then you see the dorsal roots of the spinal nerves that are going to form the coda equina. This is a dorsal section on an MRI. It's a T1 and Hayes sequence. And then you see the spinal cord here. And then as the spinal cord gets at the caudal level of the spinal cord becomes thinner. And these are here, we should find the caudal segments of the spinal cord. And then the spinal cord progresses caudally as a cord of glial cells that is called filum terminale. And then you have here all the epidural fat. On this image is also a dorsal section on an MRI, and then you identify this is the sacrum, and you see how long are the roots that form the last lumbar, and then we should have here the sacral and caudal roots from the sacral segments and caudal segments. And here you identify the spinal ganglion on a dorsal root. So as you see, all these roots are forming the coda equina. So now what we have done is to cut transversally the spinal cord at the level of the conus medullaris. When you see an MRI, you see the spinal cord, okay? And then you see these structures here, and these are the roots of the nerves that form the coda equina while they are still inside the single sleeve that surrounds the spinal cord. Here you have it. This is the dura mater and the arachnoid underneath. And then you have all these roots of the spinal nerves that are going to form the coda equina. As these are the proximal portions of the roots, they are inside a single sleeve of dura and arachnoid. However, the distal portions, they have independent surroundings of meninges, one for the dorsal root, one for the ventral root. So it means that this should be filled with CSF, and this is what we have here, filled with CSF. The epidural fat is externally located. So this is why when we do inject contrast media and we do radiographies, we see these lines here that correspond to the roots of the coda equina. And this is because the CSF is inside these proximal portions, okay? And also they extend at the distal portions because there is still subarachnoid space, but independent for the dorsal and the ventral roots. This is a sagittal section of a dissection. We are at the level here, we are at the level of the sacrum, L7, L6, L5. And then you see the spinal cord getting thinner. This is the conus medullaris. And it's surrounded by all these proximal roots that form the coda equina. Here you have, this is the dura mater and the arachnoid, dura mater and the arachnoid. And here we have the subarachnoid space with all these connective trabeculae here connecting the pia mater and the arachnoid membrane. This is a video, but to show the same structures. Okay, the conus medullaris This is L7, this is L6. So the spinal cord gets up to L7, L6, and then all these structures belong to the proximal portions of the roots that form the coda equina, surrounded by the subarachnoid cavity with CSF. Here, this is a dorsal durectomy to identify the spinal cord from the cervical level. And as you see, this is the dorsal funiculus that is quite wide at the cervical level. And as you see, all the small roots that are going to join to form a dorsal root of a spinal nerve. And as you see, the dorsal funiculus gets thinner when it gets to the thoracic level. See, it gets thinner. This is the dorsal funiculus here. We'll talk about that in other talks. You see, this is the spinal root of a spinal nerve. And then we are getting to the lumbar level. And we are reaching the lumbar intumescence. Here we have the lumbar intumescence. And at this level, the dorsal funiculus gets wider again. And then we progress caudally up to, this is the conus medullaris. And then you see all these roots that are the proximal portions of the roots of the spinal nerves that are going to form the coda equina, that are inside the same dura mater and arachnoid membranes. Okay. And here they are located inside the sacrum. Let's have a look on MRIs. Okay, we are at the cervical level. This is the axis, C3, C4, C5, etc. We are going to enlarge this area here. And what do we identify? We identify the spinal cord here. We are at the level of an intervertebral disc. This is the nucleus pulposus. And as you see, this layer here is the dura mater plus the arachnoid membrane. So on top of that, we have the epidural fat in the epidural space. And underneath, we have the subarachnoid space filled with CSF, cerebrospinal fluid. If we look at a lumbar level, we see the same. We see the spinal cord, and as we see, there is more CSF, more subarachnoid space, the dura mater, plus the arachnoid and the epidural fat. And as previously at the cervical level, most of this epidural fat located dorsally in the spinal cord is at the level of an intervertebral disc. Okay, there is more epidural fat dorsally than ventrally. Here is the same, epidural fat, epidural fat. Here at the C1, C2, and C3 spinal segments, C3 is inside the axis, the second cervical vertebra. The subarachnoid space is bigger, so we see more signal of the CSF. Okay, and this is because the dura mater is closely attached to the floor of the vertebral canal. An important feature here is the subarachnoid space, and as you see, it surrounds the proximal portions of the roots of the spinal nerves that form the coda equina. Here is the epidural fat. Okay. So this space here is the dural sac. There's a space here. Here inside we should have the filum terminale, and here ends the dura mater that continues caudally, forming a dural ligament that attaches to the caudal vertebrae surrounded by epidural fat. So having reached this point, is important to notice a very important feature. In this drawing, I have tried to draw, this is the cranial cavity and this is the vertebral canal. Look, the outermost layer is bone. So it's a vertebra here, or is a bone that forms the cranial cavity. The layer that is underneath is part of the dura mater, because the dura mater has two layers. Okay, one is a periosteal layer of the dura mater, is the most outermost layer of the dura, and a second layer underneath is the proper dura mater, is the inner layer of the dura mater. These two layers are separated at the vertebral canal, and this space is the epidural space. However, inside the cranial cavity, both layers of dura are attached one to the other. They just detach to locate the venous sinuses or arteries, okay? And this is why the venous plexus that runs in the vertebral canal is in the epidural space, because it's a continuation of the sinuses, the venous sinuses that are in the cranial cavity, and these are located between the inner layer and the outer layer of the dura mater. So in this dissection, we are removing the encephalon from the cranial cavity. This is the floor of the cranial cavity. And what you see here, this is the inner layer that is attached at the level of the foramen magnum, but it's also these two layers, the inner layer and the outer layer of the dura, are also attached at the level of the floor of the vertebral canal, at the level of the atlas and axis. And as you see how they are attached, these two layers. However, then in the vertebral canal, in the rest of the vertebral canal, they are separated. So in these transverse sections, now we can identify things. This is at the level of C1, okay? And what do we see? We see the spinal cord with the dorsal and ventral roots that are going to form a spinal nerve. And this big space is the subarachnoid space between the arachnoid and the pia mater. You have to remember that the arachnoid is attached to the inner layer of the dura. So we have the dura mater, the inner layer here. In the next image, you see this big subarachnoid space, right? With the roots, the dorsal root, the ventral root. And then you see the dura mater. But you have to remember that the dura mater this is the inner layer. The outer layer of the dura mater is the periosteal layer. And as I have said before, we are at the level of the axis here. So the inner layer and the outer layer of the dura remain attached one another at the floor of the vertebral canal. On the lateral aspects, we have the plexus venosus, vertebralis, internus, ventralis. This is more caudally in the cervical portion, spinal cord, right? And the subarachnoid space here with trabeculae that run between the pia and the arachnoid membranes. And again, dorsal roots, ventral root. And as you see, the proximal portion of the dorsal and the ventral root are inside this layer of dura and arachnoid that surrounds the entire spinal cord. However, at the distal portions, they form, the dura and the arachnoid, they form single sleeves, one for the dorsal and one for the ventral root. Okay. Again, this is the epidural fat, and usually most of the epidural fat dorsally located is located at the level of the intervertebral disc. We progress more caudally. We see the spinal cord here, the subarachnoid space, and again, the dura mater, the inner layer. Remember that the outer layer is here. So all this epidural fat is located in the epidural space that is boundary limited by the outer layer of the dura or periosteal dura and the inner layer of the dura mater. These are the big plexus vertebralis, internus ventralis. We'll talk later on about that, the epidural ligaments. Again, important again, spinal cord, subarachnoid space, right? Subarachnoid space. This is the arachnoid and the inner layer of dura. The outer layer of dura is the periosteal dura mater. And here is filled with epidural fat in the epidural space. And again, this is a dorsal root and a ventral root of a spinal nerve. And as you see, they are surrounded by epidural fat, but here, because this root is surrounded by dura and arachnoid for the ventral root. So we are at the epidural space here. Now we have to talk about the meningovertebral ligaments. These meningovertebral ligaments are, or epidural ligaments, are very, very important. Okay, these ligaments run between the outer layer of the dura, the periosteal layer, and the inner layer of the dura. They connect both layers of the dura mater. This is the epidural space. Okay, and they are very, very important because remember, when the cervical spine lengthens during neck flexion, okay, this is important. However, as the inner layer of the dura mater is attached to the outer layer by the meningovertebral ligaments and to the ligamenta flava, the movement is allowed within the vertebral canal for an accommodation, okay, of each position of the vertebral column. In cases where there is a disc protrusion, this mobility is somehow impeded, and then the tension of the dura mater results in pain. Remember that the dura is sensory innervated. Let's have a look. This is a transverse section at the cervical level, okay? What do we see here? This is the spinal cord inside the vertebral canal. I have removed the spinal cord, but I have left the dura mater. Okay, this is the inner layer of the dura mater, and the outer layer is the periosteal layer of the dura mater. It's the periosteal dura mater. Look, we have attachments between the inner layer and the outer layer, and they are more dense at the ventral portions. These are the epidural ligaments. At the thoracic level, we find them as well. We have here, again, in between the inner layer and the outer layer, we have these epidural ligaments. At the lumbar, they are also present, okay? You see the spinal cord, the plexus venosus, vertebralis internus ventralis, epidural fat here because we are at the epidural space, okay? And the dorsal longitudinal ligament, this structure is this one, the dorsal longitudinal ligament. And then as we move the inner layer, we identify as well these epidural ligaments. And remember, they are very important to allow the movement of the spinal cord inside the vertebral canal. Caudally, at the level of the cauda equina, they are also present, but they are as not dense as in the cervical level. Let's have a look at these ligaments. This is a transverse section at the level of C1 and C2. This is the dense of the axis. The encephalon has been removed and part of the spinal cord. And what do we see? With the forceps, we are moving the inner layer of the dura. Okay, and as you see, this inner layer of the dura is attached to the ligamenta flava. And also there are these epidural ligaments. And we identify something here in the epidural space, the dorsal internal vertebral venous plexus. Do not forget, we always say there is a ventral internal venous plexus. Okay, this is a very big one. However, there is a plexus as well, dorsally located. This is why we say ventral, because there is also a dorsal one. This dorsal internal venous plexus, it is easy to identify at the more rostral or at the first cervical spinal vertebrae and spinal segments. Again, you see this is the inner layer and the outer layer of the periosteal layer, and all these attachments are the epidural ligaments. These are very, very important. And also this inner layer is also attached to the ligamenta flava here. Remember, they are very important to allow a proper movement, and they may be responsible for pain. Again, you see the inner layer and the outer layer is the periosteal layer, and these are the attachments. The attachments here, the epidural ligaments, and also the attachments to the ligamentum flavum. Okay. More images to identify these epidural ligaments here. Again, in a video where you can see in movement the inner layer, the dorsal venous plexus, here the attachment to the ligamentum flavum, and here you see all these epidural ligaments and part of the plexus. In the epidural space, this is the dorsal one. These vessels belong to the dorsal. And as you see, all these attachments between the inner layer and the outer layer of the dura mater. Of course, ventral, we have the plexus venosus vertebralis internus ventralis here. And there is also attachments between the inner and the outer layers of the dura mater ventrally. You see, there are these small connections. Again, in this image, you see, this is we are moving the inner layer, and all these attachments between the inner and the outer layer. Okay. Now, we are going to check that in movement, in flexion. We are at the lumbar, spinal cord. Okay, here we have the lumbar intumescence. So when the column flexes, the spinal cord moves inside the vertebral canal. And in order to avoid problems and lesions, both layers of dura, they have to be connected. And this movement of the spinal cord is more evident at the cervical level. As you will see, it has been sectioned to see the movement of the spinal cord inside the vertebral canal. You see how it moves. Of course, there is not encephalon here, but never mind, identify the movement of the spinal cord inside the vertebral canal. As we do not have the encephalon attached, the movement is bigger. But this is to show you that we need connections between the two layers to avoid lesions. Okay, so we are going back to the MRIs. So we see the spinal cord coming from the foramen magnum. So we are at the cervical level, and surrounding the spinal cord is the subarachnoid space filled with CSF. And as you see, here there is not epidural fat. Why? Because at the floor of the vertebral canal in this level, both layers of dura are attached one another. But dorsally, there is these attachments, okay? Dorsally, there is attachments as well. As you see, the attachments here, there is a small epidural space, but remember, these are attachments at the level of the ligamenta flava, okay? Again, we go back to dissection. This is a sagittal section. This is the occipital bone, the atlas, the dents of the axis, right? And now what we are going to do is to remove the encephalon and part of the spinal cord. And now with the forceps, we are moving the inner layer of the gyramata. Okay, we are moving it. And we discover something here that is very important for you to remember. Remember that the plexus venosus vertebralis internus ventralis is a pair structure, one on the left and one in the right. It's a big vein without valves and muscular coat. And this plexus extends rostrally and enters, or leaves, in this case leaves, the cranial cavity, forming what is known the basilar sinus. And at this level, caudal to the occipital bone, there is the ventral interbasilar sinus. This is the ventral interbasilar sinus. This is for you to remember when you do surgery at this site. Again, we see the epidural ligaments. You see in this dissection the epidural ligaments between the inner and the outer layer of the dura. More images, okay, where you see the concentration of epidural fat dorsally at the level of an intervertebral disc with the nucleus pulposus here and the annulus fibrosus. Again, we move caudally and we see the supraarachnoid space, right? This is the supraarachnoid space. The epidural space is located outside the inner layer of the gyramata. It is important, as we were saying, there is fat here, the epidural fat, you see the epidural fat, but also, look at this paper, these fat cells are also abundant in the dura that forms the sleeves around the spinal nerve roots, but they are not embedded with the laminas that form the gyramata of the dural sac. And this is important because they could have a greater impact on nerve roots than drugs stored in the epidural fat, okay? This is important for the clinicians. Now, we are going deep. We have removed the dura. Here is the dura that has been sectioned. This is a spinal nerve, okay? And with the roots that are going to form the dorsal root, okay? And as you see here, I haven't removed the arachnoid yet. So the arachnoid is this thin membrane that is bright, that is surrounding the spinal cord and the roots of the spinal nerves. When we move it, we see the arachnoid. This is the arachnoid, and these are the trabeculae, or the thin connections between the pia mater and the arachnoid. And as you see, there is a root vessel, okay, running with a root of a spinal nerve. Again, the dura, the arachnoid, well, this is the subdural space. Remember that the arachnoid in a live animal is in contact with the gyramata. This is because the pressure of the CSF in the supraarachnoid space pushes the arachnoid towards the gyramata. So the subdural space, it is a virtual space. So here we are in the supraarachnoid space. Here we are at the level of the cauda equina, and it's nice to identify all these strands of collagen fibers between the pia and the arachnoid membrane, okay? Remember, again, that we are at the proximal portions of the roots of the spinal nerves that form the cauda equina. So these are surrounded by the same sleeve of dura and arachnoid, okay? So we have all these strands of collagen fibers in the supraarachnoid space. another important structure that we have to talk about it is the tentacle ligament. The tentacle ligament is a thickening of the pia mater, okay, on both sides of the spinal cord, and it extends from C1 up to L6, L7. And you see, this is the tentacle ligament that has processes that in between the spinal nerves, they cross the arachnoid and they reach the dura mater. They attach to the dura, to the inner layer of the dura. Here you see these are the roots, the roots of the spinal nerves, a dorsal root and a ventral root, a dorsal root and a ventral root. As they go out, you see they go in a single sleeve. And here there are the attachments of the tentacle ligament. Look at this video. This is at the cervical level, and we have opened the dura mater and the arachnoid, and as you see, this cord that runs on the lateral aspect of the spinal cord is the tentacle ligament that has dentations, okay, that between the spinal nerves, they attach to the dura mater. You see, these are the attachments. These are the attachments. These are the attachments. Another concept is the septum posticum. What is that? Look, in this drawing, what I have drawn is the spinal cord, okay, that is surrounded by the arachnoid and by the dura mater, okay. We know that there is the tentacle-like ligament that runs between the pia mater and the dura mater. It is located on both sides of the spinal cord. However, there is an attachment between the pia and in the subarachnoid space, dorsal in the spinal cord. And it has been studied in humans and runs between the cervical level and the caudal level. Okay, so in dogs, what do we have? This is a section, okay, between C1 and C2 cervical spinal segments. You see a big subarachnoid space. Remember, at this level, we have a big subarachnoid space with all strands of collagen fibers inside the subarachnoid space between the pia mater and the inner layer of the dura. And at this level, you see, we have discovered these structures on both sides. They are quite short. They are just located at this level. And I have no explanation for it. Maybe it's to allow the circulation of the CSF at this space. So as we run caudally, C3, C6, C8, etc., what do we discover is these structures that are strands of fibers that at some places are more dense, and they run between the pia mater and the arachnoid. And this is the septum posticum. Some places, in some places they do not exist, and in other ones, as in this one, or in this one and L7, look how dense is these strands of collagen fibers that are known as the septum posticum. And this may be important because, as you did in these papers, they may be the cause of the cyst, okay? Okay, cyst, this is from human anatomy, but also in veterinary, we see that some cysts, there is accumulation of CSF, sometimes usually located, but not always, on the dorsal aspect of the spinal cord. And maybe the cause is the septum posticum. And remember that the septum posticum at this level is quite big. Okay, this strands of collagen fibers is quite dense in some places. So maybe it's the cause of the cyst in the CSF. Now we are going to talk about this, this place here. It is between the occipital bone, the atlas, and the axis. Okay, this is this joint here that has relevance in clinics. Okay, between the atlas and the occipital bone, there is a membrane, right? This is the dorsal atlanto-occipital membrane. The same is between the atlas and the axis. Okay, the dorsal atlantoaxial membrane, reinforced with ligaments, the lateral atlanto-occipital ligament and the dorsal atlantoaxial ligament. And why is important? Because it's the site where we do the Csf Taps. When we do that, we locate where we want to do the puncture, okay, the Csf Tap, okay, here. So we are going to go through the occipito-atlanto membranes, through this membrane that runs between the occipital bone and the atlas. It's between the atlanto-occipital membrane. And sometimes, you know, it bleeds. And the question is, where is this blood coming from? Let's have a look. This is the axis, the atlas, the occipital bone, and the encephalon inside the cranial cavity. What we do in a Csf tap? We are going to remove the axis. We identify the spinal cord here. We've removed the atlas now. And here is where there is the cisterna magna, the cerebellomedullary cistern. Okay. This is the second spinal nerve, and this is the first spinal nerve here. Okay. This is the cisterna magna, where we want to go. And now we are going to focus in this area here. Okay. Here you have, on the left side, you have a dorsal craniotomy and a part of a dorsal laminectomy. I have removed the atlas. Here the occipital condyles, the cerebellum, both cerebral hemispheres surrounded by the meninges. And look at this. What do we have here? We have structures that may be damaged in a Csf tap. Here is the C1 spinal nerve. So when we are opening the atlas dorsally, we see these adherences here. And also here we have between the atlas and the axis. Let's have a look at this video. We are going to remove the axis, and then what do we identify? Adherences. And we have talked about that. Remember that the inner layer is, and there is an inner layer and an outer layer of the dura, where there is the epidural space with epidural fat. However, the inner layer is closely attached to the outer layer at the level of the ligamenta flava. Okay. And here. And at this place here, look. And I stopped the video just to show you that there is a structure here. There is the interarquated branch. There is a vein. There is a big vein here. In the next video, look at the atlas. Remove the atlas, and what do we identify? We see the plexus venosus, vertebralis, internus dorsalis, this is the dorsal plexus here, and C1, okay, and then this, the first interarquate branch. This is the dura mater, inner layer of the dura mater. The first cervical spinal nerve, the second cervical spinal nerve. You see the second segment of the spinal cord is inside the atlas. And the first is between the occipital bone and the atlas, this here. And then we have, this is the first cervical spinal nerve, and okay. And then we have the first interarquate branch. It's a vein. So look at this. What do we have? This first interarquate branch is very close to the atlas. So if the needle runs very close to the rostral border of the atlas, it may damage this interarquate branch and it may bleed. To identify that, we have removed the atlanto-occipital membrane. We have removed it, okay? And also the outer layer of the dura. This is the inner layer of the dura. Okay. So, what is these interarquate branches? Are these ones? They are formed by the interspinal veins that run through the ligamentum flavum, and divide, okay, in these two branches that reach the intervertebral veins or the plexus vertebralis internus ventralis. It depends. At the cervical level, it reaches the plexus vertebralis internus ventralis, or at the thoracic level, they reach the intervertebral veins. Remember that these they are used to be absent caudal to T9, these interarquate branches. This plexus vertebralis internus ventralis receives blood from the spinal cord, but it's also connected with the ventral external vertebral venous plexus, okay, through the basivertebral veins that fill the body of a vertebra. You have a corrosion cast, and you see this is the plexus venosus vertebralis internus ventralis. This is a dorsal view, and then you have, this is an interspinal vein, and the interarquate branch. This is at the level of T1, and you have other ones here, other ones, and interspinal veins. In a closer look, you see that this interarquate branch is reaching the connection between the plexus venosus vertebralis internus ventralis and the intervertebral vein, because we are at T1. Again, in these sections, you see that this is the interarquate branch, okay? Interarquate branch, and underneath you have the epidural fat, okay? This is filled with fat. So remember, when you make a Csf tap, you go through this membrane, okay, the atlanto-occipital membrane. However, underneath, we have the outer layer of the dura, and then we have, in very close to the rostral border of the atlas, we have the first interarquate branch between the occipital bone and the atlas. Sometimes you can identify these interarquate branches on MRIs. Okay, you see, this is it. This is one of interarquate branch here. Okay. Again, we go back to the dissection room. A sagittal section, okay, the encephalon, the spinal cord, the occipital bone, the atlas, the dens of the axis. The meniscoid is a fatty structure, okay, to allow a better joint to. And then you see, this is the dura mater, okay, this, remember, this is the inner layer attached to the outer layer of the dura mater. And you see, look at this, we have, this is the atlas, this is the first interarcuate branch, the second, and so on, okay? And again, you see the first interarcuate branch here, okay? And yeah, in close image, you see the inner layer, the outer layer of the dura, and the first interarcuate branch. This image corresponds, okay? Right. So what else do we have in the epidural space? We have the plexus venosus, vertebralis internus ventralis. On an MRI, you see this is the plexus that runs on the floor of the vertebral canal and on both sides of the dorsal longitudinal ligament. This is the dorsal longitudinal ligament. This is a ligament that opens and attaches to the intervertebral disc and also to the floor of the rest of the floor of the vertebral canal. At the cervical level, this plexus is formed by, this plexus is formed by two big veins that they lack valves and they do not have a muscle coat. And here at the cervical level, they are quite big, okay? And they are in the epidural space. Okay, again, we see at the level of the first thoracic, you see the spinal cord here, the subarachnoid space. Here we have is an artery, the ventral spinal artery is here, okay? And then you see the roots of spinal nerves, the dorsal root and the ventral root. And remember, this is filled with epidural fat. This is filled with epidural fat. This is the subarachnoid space with CSF. And here we have also in the epidural space between the inner layer and the outer layer of the dura, we have the plexus venosus vertebralis internus ventralis. That gets smaller at the lumbar level. So remember, this plexus venosus is formed by two big veins, okay, that run on the floor of the vertebral canal. And what I have said before, this plexus is connected with the external ventral venous plexus through the basivertebral veins. Here there is a corrosion cast where you see this one of these plexus, venosus vertebralis internus ventralis, and here you have the basivertebral veins, and here is all these veins that fill a vertebral body. Remember, here the bone marrow produces blood. So when in a dissection, when we remove the spinal cord, we clearly identify, okay, the plexus venosus vertebralis internus ventralis that, remember, that is bigger at the cervical level. So again, remember, this is the plexus venosus and that runs on both sides of the dorsal longitudinal ligament. Look, this is the dorsal longitudinal ligament, okay, that widens at the level of the intervertebral disc, okay? This is the dorsal longitudinal ligament, okay? On a transverse section, you can see the dorsal longitudinal ligament, and on both sides, the plexus venosus vertebralis internus ventralis. Remember that from these two veins that form the plexus venosus on the vertebral aspect, on the ventral portion, okay, ventralis, the plexus venosus vertebralis internus ventralis, there are connections on dorsal and ventral to the dorsal longitudinal ligament, and there you have them. These are the connections between both veins. Okay. Okay, what is going to happen at the cervical level? Look, When you do a transverse section on MRI, this is the atlas, right? This is the dense of the axis, right? You see the spinal cord, right? This is the subarachnoid space. We are between C1 and C2 cervical spinal segments. And then you see that, okay? These structures here, this is the basilar sinus. And the basilar sinus run in the condyloid canal. Sorry. This is one condylus, this is the foramen magnum, right? We see the foramen magnum caudally, okay? It's a caudal view. So what is going to happen? As you see, the plexus venosus vertebralis internus ventralis running on both sides of the dorsal longitudinal ligament on the floor of the vertebral canal. What it does, it changes its position from a ventral position to a lateral position, because it has to move, because it has to continue through the condyloid canal as the basilar sinus. Okay, this is important. So it changes its position from ventral to lateral. As you see in this dissection, the atlas has been opened, the occipital bone has been opened also. You see the spinal cord, surrounded by the outer dura, the inner dura, and here is the plexus vertebralis internus ventralis that changes its position to a lateral position in order to continue as the basilar sinus through the condyloid canal. And again, you see that this is the plexus that is located ventrally and changes its position at the level of the axis in order to run on a lateral aspect through the condyloid canal to become the basilar sinus. Okay, this is the ventral basilar sinus that we have talked before. Thank you very much.